Welcome everyone to another topic of chromatography. That is, today we will discuss about the affinity chromatography. This affinity chromatography is very interesting topic since the separation is mostly based on the specificity between the molecules and the ligands that is attached to the particular gel or the matrix. Right. So this affinity chromatography mostly separates the compound based on the specificity of a molecule to binds with the matrix. So we have two kinds of specificity, that is the biological specificity we can utilize as well as the chemical specificity in the order to separate the biomolecules, right? So when we see the Pitts principle, we have to understand first thing is what is the matrix. Okay, so this matrix on activation, that means onto the matrix we will attach the ligand, that is this particular portion. Okay, so this is it is the matrix and that is the ligand. So matrix ligand interaction is the most important things in the affinity chromatography. So to the matrix we attach the particular ligand molecule, and then once this is ready we will add the sample, right? So sample would be of a mixture of different molecules. Okay, when we add these different molecules onto the column where the matrix is attached with the ligand, only that particular samples or the molecule will attach to the ligand which has the specificity towards the ligand molecule. That means of different structure, only the one will bind which has the similar structure with the ligand. So out of this, only these particular samples or the molecule it can binds with the ligand. Here you can see this, right? So rest of the unbound molecule will be removed out of the column. So and after this, what is interested to us, it is our sample. Okay, so we will washing will remove the unbound sample, and finally step is what, it is the elutions. Okay, so elution is the process of separation of a sample from the ligand. So we can change the buffer system so that the sample molecule get detached from the ligand and get finally isolated separately. So this is what exactly the principle of affinity chromatography. This whole process is mostly based on the basic principle of matrix and the ligand interaction. Okay. So matrix is generally a chemical compound. Mostly we use it as a stationary phase to which we attach the ligand. Okay, so when the matrix and ligand bind, they produce a matrix ligand complex. That is, there is a binding of the ligand with the matrix. So matrix ligands complex, to this we will add the sample, right? So sample which is having the specificity with the ligand molecule, they only binds, right? So this is called the immobilized sample. Now this particular sample which is attached to the ligand, it is finally cannot be separated by the elution process. Okay? So this is what exactly a matrix ligand interaction. So we have to see what is matrix, what is ligand. Matrix it was mostly we use as stationary phase. They are all mostly type the gels. Okay, gel kind of mostly agarose or the mostly polysaccharides. Right? Onto this particular agarose or the polysaccharide gel, there are certain side chain groups, such as amino group, hydroxyl carbonyl group, and the particular ethyl groups. Okay? They are located at their particular structure, which actually serve as a binding site for the ligand. That means these particular side chain groups are helpful for the attachment of the ligand to the matrix. So matrix generally is actually give the particular area, surface area, so that the molecule can bind to the matrix, right? So matrix generally it only increase the surface area so that the molecule can bind. So what is ultimately we understand from this, matrix are the gels. Onto that gel, particularly side chain groups like this, they are attached, okay, so that the ligand can bind to this side chain. The use of matrix so that it increase the surface area, therefore the molecules can bind to the matrix molecules. Okay. See, cellulose we can use. Okay, cellulose used mostly for the DNA isolation. Polysaccharides the mostly polyacrylamide. It is used again. It exists both in the gel and the beads form. The beads form not sufficiently porous. That means therefore we cannot utilize that. Okay. So mostly the gel form of the polyacrylamide we use for the stationary phase. Right? And finally the agarose. Okay. Agarose again is used for the DNA isolation. So these three types of particular matrix molecules we will use as a stationary phase, onto which we will bind the ligand. Right? So ligand is again, what ligand is? The chemical compounds. Okay, small group of a chemical which is attached to the matrix. Right? Ligand should interact with the desired molecule by forming a temporary bond. Okay? The interaction is a temporary. The molecule and the ligand interaction is for a short while, so that we can separate the sample molecule from the ligand. Therefore, the bond between the ligand and the molecule should be of a temporary. Right? So ligand molecule complex will remain in the column, and then rest of the unbound things they automatically remove. Right? So ligand molecule complex, okay, they dissociate. That means we have to separate the samples, okay, of our molecule just by changing the pH of the medium. So this pH increases the ionic strength that dissociate the sample molecule from the ligand. Right? So in this way, the specificity generally we define. Okay, three things we require. One is the matrix, second is the spacer arm, third is the ligand. Okay, so this is the matrix. Okay, this is the spacer arm, and this is the ligand. Right? So to the matrix we use the spacer mostly the particular acetyl group actually, which helps the binding of the ligand to the matrix. Right? So this ligand get binds to the spacer, okay, and the spacer then they get binds to the matrix molecule. Okay? So in this way we generally make the criteria of this stationary phase. Right? So ligand is not directly binds to the matrix. Okay, so first it binds to the spacer and then to the matrix. See, in case of specificity, we can isolate the number of different biomolecules as the antigen specific to the antibody. So we can isolate the antibody. Vice versa, antibodies are specific to the antigen, so we can isolate the antigen also. Right? Substrate are specific for the enzymes, so we can isolate the enzymes also. Right? To isolate the DNA, we need the DNA binding site, or generally the protein, that is the histone. Okay, so DNA is bound with the histone, so we can use the histone as a ligand in the order to isolate the DNA. And the hormones, mostly all the receptors to which the hormone binds. Right? So we use the receptor as a ligand. Okay, so in this way, this is all different types of example by which we can isolate the particular biomolecule by using their counter ligand, okay, or their specificity partner, so that we can isolate the different types of biomolecules. So this is exactly the process of affinity chromatography. After preparing the matrix with the ligand, that is into the column, okay, we add the sample of a different mixture. Right? Then from the different mixture of a sample, only the sample which having the specificity with the ligand molecule, then only bind. Right? So out of this particular different mixture of sample, only this particular sample will get bind because it is having the specificity with the ligand molecule. Okay? So here this is the sample of interest which is bound to the ligand molecule, and rest of the unspecific samples, okay, they are eluted out of the column. Okay, after washing. Okay, so they cannot bind to the ligand, so they easily remove from the column first. And after that, in the order to remove these particular sample which is attached to the ligand, we have to carry out the elution, where we will add the buffers of a different ionic strength, which helps in the detachment of the sample from the ligand portion. Okay, so these particular samples they actually get detached from the ligand, यहाँ से detach हो जाएंगे सब, and then finally they move out of the column and then get separated. So this is overall the procedure or the working mechanism of the affinity chromatography. First is the addition of the sample which binds with according to their specificity, then separation of unbound samples, and finally the elution of a sample of interest. So these are the three main basic processes of the affinity chromatography. Most important thing what we have to keep in mind here, it is the interaction of the ligand with the sample molecules based on their specificity, right? When we see the application of this, okay, the most advantage of this is what is this, the purification. That is, it can purify the samples from the different mixture. Okay, they also reduce the amount of a substance into the mixture. They also purify and concentrate the enzyme solutions. So affinity chromatography is mostly used for the purification and the isolation of the different types of enzymes by keeping their substrate as a ligand into the column. So in this way we can isolate the different types of biomolecules, right, based on the specificity. So affinity chromatography, mostly it actually separates the component based on their specificity, like enzyme, receptor, ligand, and the proteins and the nucleic acid, etc. Okay, their substrate and the different counterparts. So in case of its component, we will have a column. Okay, column generally it is made up of glass, is filled with the matrix molecule. So this matrix molecule is most of particular different types of gel which have the characteristic like it should be chemically inert, should have good flow properties so that the buffer can easily flow, and have the different types of functional group that can attach to the ligand. Types of matrix, they are mostly gel, agarose, polyacrylamide, polystyrene, cellulose, and the silicas. Right? So these particular matrix molecule is attached with a particular spacer, mostly the acetyl group, which helps the binding of the ligand to the matrix. The spacer is very important, right? In case of its working, generally the different mixture component sample is added into the column, and the most specific sample which is having the specificity the ligand can bind to the ligand. Rest will be easily removed. Okay, and then we can remove the sample of interest from the ligand by changing the pH or having the different pH with the different ionic strength. It can be removed from the column and purified, right? So, and to remove again by the different kind of a method, like we can add the competitive inhibitor to the sample. So the competitive inhibitor bind to the sample and remove, and this competitive inhibitor from the sample again can remove by dialysis or change in the pH or the change in the ionic strength of buffer. So we can have the different example of ligands in the molecule that is interact or to separate. So for example, AMP, it can use as a ligand to separate the NAD dependent enzymes or the kinase. ADP as a ligand we can use to separate the NADP dependent dehydrogenase. Calmolidine as a ligand we can separate the calmolidine binding enzyme. Avidin we can use to separate the biotin containing enzyme. Nickel mostly we use to separate the protein with the histidine tag. Lysine as a ligand we can use for the purification of the RNA molecules. Okay? So this process of affinity chromatography mostly consists of 3 to 4 steps: equilibration, sample addition, washing, and the finally elutions. Okay? So first step is what is the equilibration, where the spacer molecule and the ligand molecule it is equilibrated with the stationary field. Then we add the sample. Now the sample based on their specificity they get attached to the particular ligand, right? So the specificity is more important so that they can attach to the ligand molecule. And finally we do the separation. For the separation we will add the buffer molecule that helps the detachment of the sample from the ligand. So this pH or the buffer molecule dissociates the samples. So this is very interesting affinity chromatography, which is mostly based on the separation of a biomolecule with the specificity of a molecule with the specificity of the ligand. Okay? So this is all for today. We'll continue with the another topic. Thank you very much.